Fox here and is listening in too. So thank you for making this possible, Jerry. And the award this year goes to Emma Holmes. She is awarded this for her pioneering studies of auditory perception and speech processing. And to introduce her, we'll have and talk about her work a little bit will be Dr. Timothy Griffiths.
Thank you so much, John. It's my great pleasure today to introduce Dr. Emma Holmes, who works on what might be characterized as the hard problem of auditory neuroscience, how the auditory brain allows listeners to appreciate the natural sound world. Her work combines an impressive array of techniques, but really has a single common theme that is central to our field, the mechanism for hearing speech and noise. Emma developed EEG expertise during a distinguished career as an undergraduate in Oxford. She then went on to do a PhD with Quentin Summerfield at York, during which time she identified EEG signatures for the analysis of speech in noisy environments. Her postdoctoral work was carried out in Canada with Ingrid Jonsrud and at UCL in the UK with myself and Carl Friston. During her postdoctoral work, she has uncovered both low-level bases for speech and noise perception, fundamental grouping and streaming mechanisms, and also high-level influences on speech and noise perception, such as voice familiarity and task effects. She's carried out further brain measures as a postdoc, including functional MRI and MEG. In addition to those, she's applied advanced modeling techniques to define the brain systems for speech and noise listening, as opposed to the simple definition of brain networks. One of the most impressive aspects of Emma's work that I wanted to highlight today has been her determination to tackle what might be thought of as the even harder version of the hard problem, natural listening in subjects with hearing impairment. This is a fundamental scientific problem, but also has obvious, tremendous translational potential. And this was recognized by the award of a Pauline Ashley Fellowship from the UK hearing charity that is now called the RNID. Emma's current program of work that she's been carrying out as a new faculty member at UCL addresses the effects of hearing loss on real-world listening and the associated brain mechanisms. In addition to her scientific work, I also wanted to point out the great effort that she makes to reach the deaf community that informs her work. And she's also supervised and mentored an impressive number of students. In short, Emma Holmes is an ambassador for clinically relevant neuroscience that will translate into better lives for patients with hearing loss. And as such, I think she is a just bearer of this award, named after this pioneer of patient advocacy for the deaf, Geraldine Dietz Fox. Congratulations, Emma.
Thank you very much, Tim, and to the organisers as well. I was asked to prepare a presentation about some of my work, so I think that should be coming up shortly. Thank you. Hi everyone, I'd just like to thank the ARO committee for the Geraldine Fox Young Investigator Award, which I feel very honoured to receive. I've been asked to give a presentation about my research in just a few minutes, so I'm going to give you a brief overview of my work here, and I'll also provide some citations on the slides where you can find out more information if you're interested. My research addresses the question of how we understand speech when other sounds are present, and most of you have probably experienced many times that this can be quite challenging. We know that these noisy environments are particularly difficult for people with hearing loss. It's the most common problem they report finding difficult. And it's something that's not well addressed by current interventions. Hearing aids can provide quite large benefits when people listen to speech in quiet, but in noisy environments they'll also amplify much of the background noise, meaning that it can still be very difficult to understand speech in noisy places. Another interesting clinical phenomenon is that 5 to 15% of patients who visit audiology clinics for hearing difficulty actually turn out to have normal audiometric thresholds. And these people usually report particular difficulty listening in noisy places. But we don't currently have a good explanation for why this is. Lots of previous research has focused on peripheral factors that might contribute, But understanding speech in noisy places also requires a variety of cognitive processes, such as attention. And I've been interested in how cognition might contribute to these phenomena. My research can be divided into 3 main strands. The first aims to understand how people with normal hearing use cognitive factors such as attention and prior knowledge to improve speech and noise perception. The second aims to understand how the cognitive processes engaged during speech perception are affected by hearing loss across the lifespan. And the third aims to understand why some people with no detectable hearing loss have substantial difficulties with speech and noise perception. So today I'll give you a brief overview of some work I've done to address these questions. There's a wide body of literature showing that various cognitive factors affect speech and noise perception, and throughout my time in various labs I've had the opportunity to study particular factors in more detail. One of those factors is being familiar with the voice of the person who's speaking. I became interested in voice familiarity when I went to work in Ingrid Johnsry's lab in Canada as a postdoc. A previous study by Ingrid and colleagues investigated familiarity with a spouse's voice in couples who had been married for more than 10 years. They presented 2 CRM sentences simultaneously and asked participants to report a target sentence that was either spoken by their spouse or by one of the other spouses in the study who the participant had never met. They found a 10 to 15% improvement in speech intelligibility when participants were asked to report words spoken by their spouse than by an unfamiliar person. And for various reasons I don't have time to go into, this can't simply be explained by attentional salience or by a bias towards reporting words from the spouse's voice. In follow-up work, we found that people don't need to know someone for as long as a spouse to get this intelligibility benefit. Very consistently, we found a similar magnitude benefit when participants report sentences spoken by their friend. We've even found a similar effect when we train participants to recognise novel voices in the lab. We found a small intelligibility benefit after 10 minutes of training, but we found a large benefit similar to that for naturally familiar voices after just 1 hour of training. Prior to this, studies had shown that voice training in the lab is possible over several days, but we were quite surprised to find such large improvements to intelligibility after such a short duration of training. And we did a variety of other studies that showed that familiar voice effects are greatest when the background is an intelligible competing talker compared with an unintelligible talker or noise. We also found that spatial patterns of activity in left posterior STG, MTG, and planum temporale are more robust for familiar than unfamiliar voices. We also found that improved intelligibility of familiar voices relies on a different combination of acoustic cues than familiar voice recognition. Even to the extent a familiar voice can be more intelligible than an unfamiliar voice, even if it's not explicitly recognised as being familiar. During my PhD working with Quentin Summerfield, I studied how a different cognitive factor affected speech intelligibility, and that's preparatory attention. We sat participants in the centre of an array of loudspeakers in which they heard 3 different phrases spoken by 3 different talkers from different locations. And we told participants which talker they should listen to in advance by presenting a visual cue. We found that when participants had more time to see the cue and prepare their attention before the talker started speaking, they performed better. We found reaction times got progressively better as participants had longer to prepare for the target talker. And we found quite similar patterns of results for spatial cues and gender cues. More recently, I've been modelling these situations in collaboration with Carl Friston at UCL to help understand the mechanisms underlying this benefit from preparatory attention. Crucially, I've also utilised this paradigm to ask how preparatory spatial attention is affected by hearing loss. We found that preparatory spatial attention could be measured using EEG, both in adults and children with normal hearing. However, we found that children with mild to moderate hearing loss showed no evidence of preparatory spatial attention. In addition, this wasn't restored when the children used their hearing aids. My current work builds upon this finding by asking whether adults who experience hearing loss later in life show similar effects, and I spoke about this in the Aging Podium session yesterday. But although people with hearing loss show weaker preparatory spatial attention, we found that they benefit from semantic context like people with normal hearing do. We found that adult hearing aid users were able to report speech and noise much better when the sentence was the same topic as the previous sentence, which is shown by the darker bars here, than when the topic changed, which is shown by the lighter bars. We also found that this led to reductions in self-reported effort. Overall, the benefit from semantic context seemed to be independent of the benefit that they got from changing their hearing aid settings. Finally, I've been interested in why some people with normal hearing on clinical tests report particular difficulty understanding speech in noisy places. And when I was a postdoc at UCL working with Tim Griffiths, we hypothesised that difficulties understanding speech in noisy environments might relate to auditory grouping. In other words, the ability to separate target and background sounds. So we developed new tests of figure ground perception that assess auditory grouping. The participant has to follow a pattern of tones, shown in red here, against a background of similar tones that have random frequencies. On each trial, they have to decide whether there's a gap in the figure or not. Some of the figures stayed the same frequency over time, whereas others changed frequency over time, like the formants in speech. And the short version is that both tasks help to predict individual differences in speech and babble perception. And moreover, they index different portions of the variance across people. This suggests that some people might find speech and noise difficult because they find it difficult to pick out frequencies that remain stable over time, whereas other people might instead struggle to follow a target stimulus that changes frequency over time. Using neuroimaging, we found that this shared variance likely occurs at the earliest stages of auditory cortical processing, in TE 1.0 and 1.1. We also found that a patient who experienced damage to auditory cortex after a stroke had deficits for speech and noise perception, for music streaming, and also for same frequency figure ground tests. And this supports a cortical substrate for the shared variance. It also implies that these figure ground tests might assess domain general grouping processes that are relevant to both speech perception and music perception. So to summarise, I've looked at how cognitive factors such as voice familiarity and preparatory attention improve speech intelligibility in people with normal hearing. My work has shown that children with hearing loss display weaker preparatory attention, which isn't restored by hearing aids, and might help to explain why they have difficulty understanding speech in noisy environments. Yet older people with hearing loss can benefit from semantic context over and above the benefit they get from changing hearing aid settings. Finally, we found substantial variability in speech and noise perception among the population who supposedly have normal hearing, and some of these differences appear to be predicted by grouping processes at early stages of auditory cortical processing. I'd just like to finish by thanking all of my scientific supervisors and collaborators over the years, some of whom are displayed on this slide. I'd also like to thank the funders, particularly RNID, who are funding my current work. I'd also like to thank the mentors who have helped me along the way. There are too many to list here, but they'll know who they are. I recently joined the faculty at UCL, so if you'd like to work with me, feel free to get in touch. And thank you for listening.
Thank you, Emma. That's amazing work. Just really wonderful, wonderful work. And I guess we're kind of moving on with our Year of the Cortex here, it looks like. It's my distinct honor to welcome Christoph Schreiner and thank him for his work towards our society and towards the science that he's done. He is our 2022 Award of Merit recipient, awarded for seminal studies of central sound processing and cortical plasticity. These findings have set the basis for understanding the effects of hearing impairments on cortical function. And we have two people that will introduce him and talk about his work a little before he speaks, Dr. Daniel Pauly and Dr. Robert Liu.
Hello, everyone. Can you all hear me? All right, great. Hi, I'm Robert Liu, and I'm delighted to welcome you to this virtual ceremony honoring Professor Christoph Schreiner for his ARO Award of Merit. In this introduction, we'll try to provide a little insight into the man behind this very deserving recognition.
I'm Dan Pauly. I'd like to kick things off by first offering a bit of a historical perspective. The ARO has honored 46 pioneering researchers with their Award of Merit. Their selection of Christoph has special significance because it is the first time they have recognized a researcher who has devoted their career to studying sound processing at the level of the auditory cortex. In the BC era, that's before Christoph, the auditory cortex was not a very popular system to study cortical sensory processing and perception. Work on the auditory cortex has always been dwarfed by studies of visual cortex, but even compared to the somatosensory cortex, auditory cortex publications lagged behind. But somewhere in the late 80s and early 90s, productivity really started to ramp up as the auditory cortex became a premier system for research on neural coding and adult experience-dependent plasticity. Now, looking within the auditory system at keyword hits among ARO abstracts, we can see a similar story. The online archives of ARO don't allow me to go back into the BC era, but even looking over the past 30 years, one can appreciate that the appearance of auditory cortex as a keyword has tripled relative to other major stations for neural sound processing, which are pooled together here in gray. Now, to what can we attribute this marked uptick in auditory cortex productivity over the last 30 years? Clearly, the answer is that it's all because of Christoph and the remarkable members of the Schreiner Lab over the years. I'm kidding, of course, because the truth is that Christoph was one among a cadre of key principal investigators that really took up the challenge of unraveling the mystery of how sound was encoded at the level of the auditory cortex and how these neural representations supported auditory perception, both in the context of normal and disordered hearing. But Christoph is a superb choice as the first researcher among this group to honor with the award. To put some numbers on that statement, let's pull Christoph's trading card and have a look at his stats. In terms of scholarship, Christoph has been highly productive and impactful with 144 publications, garnering 24,481 citations and an H index of 85. Christoph is a revered teacher and mentor, having supervised 20 PhD students and 35 postdoctoral trainees over the years, has been an active member of the academic community, where he serves as vice chair for research in the OH&S department at UCSF, in addition to professional service for the ARO and other international organizations. Christoph's research has been continuously supported by the NIH since 1988, and importantly, he is by no means done. He is PI on 3 large active grants and continues to develop sophisticated neurophysiological and computational approaches to understand neural processing of signals and noise and the disruption of these normal processes following sensory neural hearing loss.
So how did Christoph reach this level of impact in the hearing sciences? In many ways, his whole life has been leading up to this. Christoph grew up in northern Germany, where frequent trips into the woods fostered a love for nature. His favorite subject in school was biology, but when he selected a professional field for training, his other passion for more quantitative and physical sciences steered him into studying physics at the University of Göttingen in 1969. He worked under the tutelage of Professor Manfred Schroeder, who was renowned for using innovative applications of math to various acoustic problems in the real world relating to speech, hearing, and concert hall acoustics. In fact, Christoph's first exposure to modulation transfer functions and auditory psychophysics came while he completed his physics master's and doctoral theses, setting Christoph on a road to using rigorous quantitative methods in the hearing sciences. Along the way, Christoph enrolled in medical school to learn human physiology, since he became convinced that psychophysical phenomena had to be understood in the context of both the biophysics and physiology of the sensory system. In 1977, with a fresh PhD physics in hand, Christoph embarked on actual physiological studies of the auditory system in his first postdoc at the Max Planck Institute lab of Professor Otto Creutzfeldt, an early pioneer in higher order visual neurophysiology who wanted to expand into audition. It was there, while he completed a doctoral thesis in medicine, that Christoph published his first highly cited paper on the thalamocortical transformation of responses to complex auditory stimuli in 1980, a few years ahead of the coming explosion in publications on auditory cortex that Dan described.
After the completion of his training in 1980, Christoph was offered the chance to lead the auditory group at Max Planck, though he was encouraged by Professor Creutzfeldt to first do a short postdoctoral stint in California before returning to Germany to take the reins. Now, after an initial crash course in American culture at Disneyland, Christoph headed to UC San Francisco to work with Mike Merzenich and a team of researchers developing a multi-channel cochlear implant. The fellowship was a success on many levels because Christoph became friendly with Marcia Raggio, another researcher on the cochlear implant research team, and their collaboration bore fruit, both academically and personally, as we can appreciate from this photo of Christoph, Marcia, and their daughter, Christina. That collaboration with Merzenich was also a massive success, and we all know that what was envisioned as a one-year fellowship wound up as something else entirely. Merzenich and Schreiner began a 30-year collaborative partnership that in many ways defined a field. producing several canonical works on the organization of sound frequency and other feature representations in the cortex, as well as the mechanisms and therapeutic applications of cortical plasticity.
If you make a word cloud of the abstracts from Christoph's greatest hits, you get a nice illustration of how comprehensively his work has impacted our understanding of the central auditory system. He has made major contributions to elucidating functional auditory cortical organization, as well as midbrain and thalamic functional organization, cortical plasticity from synapses to maps, hearing impairment effects on the cortex, and of course, complex auditory receptive fields of cortical neurons, something you will hear more about in Christoph's lecture.
Christoph has shown an abiding belief in the virtues of collaborative research. The primary affiliation listed on all of his papers is the Coleman Lab, an endowed laboratory within the department that is the intellectual home for clinicians, scientists, and researchers in the department, with an interest in the organization of the brain's hearing systems. In addition to the many seminal works published with Merzenich over the years, Christoph has also been enormously successful, a longstanding collaboration with Steve Chung, a neurologist in the department, with Ralph Beitel, with his right-hand man, Craig Atencio, and more recently, with new faculty recruits to the Coleman Lab, Brian Malone and Andrea Hassenstab. Christoph's collaborations extended beyond UCSF as well. The late Jeff Weiner, who spent his career across the Bay at the University of California, Berkeley, is widely regarded as one of the great neuroanatomists of the last 50 years, who, to the great credit of our field, happened to focus his efforts on the central auditory pathways. The collaboration between Weiner and Schreiner was truly nonlinear, 1 + 1 = 1 million kind of partnership that projected the Schreiner Lab gorgeous functional maps of sound feature representations onto the underlying lattice of neuroanatomical connections. The primary research papers are all paperweights, dense, rich, hugely informative, but the review papers are beautifully streamlined, provide a remarkable introduction to the field because they cut through the inherent complexity and introduce clear, inspiring, and testable models that have been a source of inspiration for many of us.
And it's not just his science that has been an inspiration. Another equally important contribution Christoph has made to our field is the 12 PhD and 17 postdoc trainees he has helped supervise over the past 25 years who have gone on to become faculty. Christoph has seeded multiple generations of auditory scientists in the academy. Through his thoughtful mentorship and unique blend of German seriousness and humor. Dan and I were both at UCSF in the early 2000s, along with this cohort of Coleman Lab colleagues and future professors, which includes the so-called Three Musketeers who implemented the dynamic moving ripple stimuli you'll hear Christoph talk about. Christoph has always been generous with his time, and you could walk into his office and he would be willing to put away whatever administrative work had been heaped on him to listen to you talk about your science and offer advice, sometimes playfully. And for many of us, Christoph continues to offer words of wisdom to put things into perspective, always with the long view on your scientific contributions and your life and family. Had we been able to celebrate his award in person, I'm sure we'd all be going out to a Coleman Lab alum dinner, which Christoph loves to host whenever we see each other at meetings. We'll take a rain check and look forward to toasting him in person next time. We all could not be happier to see Christoph receive this well-deserved recognition.
Congratulations, Christoph.
Okay, is it up to me now?
Whenever you're
It's up
ready,
to you.
can share
Oh,
your
I see.
file.
All right. Okay. So, okay. Well, first, let me do the sharing here. We have a proper background and get the right pointer. There we go. All right. Well, first, I want to thank Dan and Robert for the very kind and generous introduction. Really humbling in a way. But first, I want to really give my sincere thanks to the nominators and ARO for considering me for this great honor. It is truly the highlight in my professional life. But I also want to express my heartfelt delights and gratitude to all people that enabled me to get here, because this is all based on teamwork with many terrific collaborators, old and young, and we'll come back to that a little bit later. In particular, though, I want to thank my family, Marcia and Christina, for putting up with long experiments, sulking grant writing stretches, and many moments of staring absently into the ether. But you always have to provide a foundation of love and acceptance. So the topic today that I will be talking about, not quite as smoothly as in the previous presentations, but here it is the auditory cortex. Now, auditory cortex is essential for perception and cognition. Yet, despite tremendous progress in the field, a significant gap of knowledge still exists about the relationship of cortical neural activity and hearing ability in normal and hearing impaired subjects. So to bridge this gap, we have to learn much more about the function of auditory cortex. So today we will explore from several perspectives the cortical processing of one particular aspect of sounds, namely that of spectral and temporal envelope modulations. So this team, auditory cortex and modulations, has been with me for a long time, since the late 70s, actually. They were inspired by my early mentors, Manfred Schroeder and Otto Kreuzfeld.
Dr. Schreiner, I'm so sorry to cut you off. I'm not sure your slides are progressing.
I didn't advance anything yet.
I'm so sorry. Thank you.
That's all right. We'll get there in a minute. So this is sort of just a general statement. It started early in my experiences, and it's still going on, and it's still fun to do. So let's talk about the cortex in a little more detail. So here is a picture. Something's overlaying my thing, but in any case. So a prominent principle in biology is that structure determines function. Now, the structure of auditory cortex is rather complex. As you can see in the left drawing, there are lots of different cell types. There's dendritic trees with axons and different cell types, excitatory, inhibitory, and so on. You can see there's some kind of layering of the internal organization. On top of that, there are lots of inputs that really determine what cortex is doing. There are inputs from the thalamus, here the MGB, medial geniculate body, that go to different layers. There are a great amount of cortical connections that enter each of these layers. Actually, the cortical inputs make up almost 80% of what converges on a neuron in auditory cortex. Then there are all the exits, the outputs. Each layer has its own target and provides information. And there are several layers, layer 5 and 6, for example, project to subcortical stations. So there's a lot of structure, a lot of complicated connections and topologies, but the function is derived from the dynamic interactions of all these elements within and across all cortical scales. So the global aim has to be, at least from my perspective, to understand the cortical functionality across all its structural scales. So we said we would only focus on one sort of sound aspect today in today's presentation, and that is that of stimulus envelope modulations. Now, natural sounds are often broadband with dynamic changes in the spectral and temporal properties. Spectrograms like this one up here for a speech segment illustrate the diversity of spectral modulations. So that is, if you look along the frequency axis here, there are narrowly spaced energy peaks, there are broadly spaced energy peaks, and they sort of summarize the spectral modulation that are in inherent in all natural sounds. At the same time, there's also temporal modulation. If you look along the time axis, there are long segments, there are short segments, and one can simplify this approach of looking at spectral and temporal modulation aspects by projecting these properties on a sort of a two-dimensional system, one dimension being spectral modulations, the other one being temporal modulations. And at each moment in time, you can identify in artificial stimulus that we are using to explore this, what is at any moment going on with regard to spectral and temporal modulations expressed here in these little segments of this artificial stimulus that we use for our exploration. We call it a dynamic moving ripple that changes these combination of spectral and temporal moments and properties over time in a sort of pseudo-random way. Now, these two dimensions, spectral and temporal modulation, give us sort of a broad picture of what is the composition of sounds, and tell us a lot about the sounds, maybe not everything, but they capture quite a bit of information. Here's an example of speech segments that shows us, for example, that there's a wide range of spectral modulation present in speech, whereas the temporal modulation range is fairly limited. The highest power is sort of below 20 hertz. In many sounds. So, but how do we now capture how cortical neurons respond to these two dimensions of spectral and temporal modulation? So an accepted and useful way is the determination of spectral temporal receptive fields that has been done for quite a while. And how is it done? So, for example, we use the dynamic moving ripple as a stimulus. So again, at any moment in time, it is described by two parameters. What is the spectral modulation? What is the temporal modulation? And then we look for the responses. So in this case, here are some action potentials up here. And in order to find out what drives those spikes, we just average the stimuli that precede each of the stimuli. We average them up, and the result is what we call the STRF, spectral temporal receptive field, that has some characteristics here. You can see at time 0 is actually the time of the spike. So this is the spectrogram that precedes, on average, a spike. There are red areas that have high energy in the stimulus. There are blue areas that energy is below the average. Both of those conditions are necessary, apparently, to create an action potential. Now, this doesn't tell us that much immediately looking at it. What is the modulation preference of these neurons? Because that's our target today. But if you apply a two-dimensional Fourier transform to this, you get what's called the modulation transfer function that plots the stimulus energy preceding a spike in terms of what are the average spectral modulation and what are the best tempo modulation. So this gives us a fairly compact description of modulation properties of any given neuron. So the next slide gives us a little bit of an overview of the topics we will be touching on. So as I said, we want to look at different scales of the auditory system, from large to small. And so we'll start out in a bit with looking at cortical fields, particularly the primary auditory cortex. We look at organization within the field, large scale and local organizations. We look at how temporal modulation and spectral modulation change across the cortical layers. Then we move to slightly more complex themes and trying to understand, is there a better way than the STRF, the spectral average approach, to measure what a neuron is doing? We also will look at the synaptic integration properties that may lead to the cortical neuron preference for specific temporal spectral modulations. And at the very end, we'll have a quick look at groups of neurons that fire together in some kind of correlated way and see what they might be able to contribute to modulation, encoding, and processing. Okay, so let's move on with our first topic here, which is looking at auditory cortical fields. thing. Something is blocking the top of my screen, but I'm not sure whether I can do anything about it. Anyway, so auditory cortex has been known to contain many different cortical fields. They are separated out as core belt or parabelt regions. And this classification is really based on their connectivity from the thalamus and between cortical regions and other functional differences between them. Now, today we will really focus mostly on the primary auditory cortex, A1. Here, this is an example in the cat, and A1 is pretty much all mammals is organized in some kind of frequency-specific way, or tonotopic or cochleotopic way, with the response to tones on this side, on the left side, is mostly for low frequencies, and then you can see an increase in the preferred tonal stimuli from high to low. So this is a very nice global organization of A1. But on top of that, there are other local organizations that also can be studied and have been studied. In this case, there is something that is not directed to what frequencies are encoded by a neuron, but what range of frequencies are encoded. So these brownish red areas are locations where the range of frequencies that excite the neuron is very small, very narrow, and the bluish regions are locations and neurons that respond to a much wider range of frequencies. So the principle here is that A1 has global and local organization with the frequency topography with regard to spectral preference, and also with spectral integration modules that relate to spectral selectivity. Now, these modules or these little functional patches are not just functional constructs, but they also have an anatomical foundation. So it turns out so if we, how do we approach this? We made some injections of retrograde tracers in regions that are either narrowly tuned or broadly tuned, here the pink or the blue spot for the same frequency. So all these two spots prefer the same best frequency of about, let's say, 10 kilohertz. And then we look at what is the convergence of inputs to this location of injection from other cortical regions. And if you look at this here, let's look at the pink first injection, the sharply tuned region here. There are lots of inputs from the same module that converge on the location, but there's also input from another spot here, this pink region, which corresponds to another area that is sharply tuned. And then the converse is, if we make an injection in the broadly tuned region, like down here, we see that most of the inputs that converge here are either from the same module or from another one that is also broadly tuned, and another one over there. So altogether then, their cortical modules are not just functionally defined, but they have a strong anatomical foundation. And that creates a sort of an interconnected set of modules that are interleaved, that don't communicate with each other directly, but create two sort of separate streams of information that move on to other cortical and subcortical regions. Now, what does it have to do with modulation tuning, which is sort of what we are trying to study? And actually, the connection is quite strong. So the first one, the easiest, is that broadly and sharply tuned neurons, here in those two regions, narrowband and broadband regions, are direct measure of the best spectral modulation frequencies that are represented by those neurons. Neurons that are sharply tuned here, have a high spectral tuning Q value, also have high spectral modulation frequencies. And neurons that have a broad tuning have these low spectral modulation frequencies. I mean, that's a direct connection between breadth of tuning and what spectral modulation frequencies are preferred by a neuron. But that's not it alone, because there's also segregation between these modules based on temporal processing. So if you look here at a neuron from the narrowly tuned region, here's a SDRF. You can see that along the frequency axis is fairly well sharply defined. and it has a specific range of modulation frequencies. A broadly tuned neuron here from the broadband region, of course, is broadly tuned and along the frequency axis. But if you look at the modulation frequency, these two neurons differ quite clearly. This neuron here and the narrowly tuned neuron has a lower best temporal modulation frequency than what the neuron shows in the broadly tuned region, which has a much wider range it responds to with regard to temporal modulation frequencies. And that is shown here in this summary plot of population analysis. Again, this is CAT, where broadband neurons, a cumulative plot, show in general a higher modulation preference, temporal modulation preference, than neurons in the narrow band cluster or patch. So there's some kind of trade-off. In the narrow band, we have neurons that like low temporal modulation frequencies, but higher spectral modulation frequency. And in the broadband area, it's inverse. Neurons here like higher best temporal modulation and lower best spectral modulations. So there's a clear sort of segregation. And we know that these two modules have their own projection system and process sound sort of in parallel. So one of the interesting consequences of this organization, of this modular organization, it can actually be seen when it comes to the effect of electrical cochlear stimulation on cortical activity. So electrical stimulation of the cochlea for both closely and widely spaced stimulation electrodes, here is a closely spaced pair, is a widely spaced stimulation electrode pair, create activity on auditory cortex, but not equally across A1. So this is again CAT A1. What we notice is that there are regions with very low response thresholds. So the color-coded response threshold to electrical stimulation. So there are two patches of blue that respond very well to electrical stimulation, but in between and surrounding, there are regions that have very high thresholds or don't respond at all to this electrical stimulation. But it goes for both, for the narrowly spaced or broadly spaced electrodes. And not surprisingly, the area that it doesn't respond here in the middle corresponds to this narrowly tuned region if it was investigated with pure tones. So the narrowband region cortex does not respond well or at all to electrical stimulation, whereas the broadband regions respond very well. So the likely reason for that is that electrical stimulation of the cochlea creates a rather broad electrical field, resulting in poor spectral and spatial resolution, as many studies in implant patients has shown. Now, on the other hand, there's the broadband region that is being stimulated well happens to be the one that has better temporal resolution than the narrowband region. And so that may help the implant patients to rely more on temporal information to decode the stimulus that is being applied to the cochlea. Let's move on to another topic. It's another structural organization in cortex we want to have a brief look at, and that is that of cortical columns. And so a cortical column is defined as a region that has some similar properties across the cortical depth and across the layers. In A1 of many species, maybe not in all, but most species, the best frequency of neurons, so its preference of what frequency it responds to, is organized in such a columnar fashion. Here's an example of an electrode array that goes across the cortical depth, sort of perpendicular to the cortex. And if you look at the best frequencies of neurons along that electrode, you can see that actually their frequencies are quite well lined up. So this is sort of the basic function of a columnar organization across all layers. And if you look at the STRFs, on the first glance, it looks sort of similar, the STRFs here, maybe a little latency changes or so on, a little more complicated on there, but it's only when we look at the modulation properties through Fourier transform of those STRFs, we can see that there are actually quite a range of differences between different layers and different depths in the cortex. So for example, up here in the upper reaches, the the spectral modulation frequency, which is along this axis, is much more restricted for these sites than here for these middle layer neurons, which have a much wider spectral modulation range. Down here for the lower regions, we see again that there's maybe more limitation in the regard of spectral modulations, but also the tempo modulations are lower. So this difference, these differences demonstrate that different layers likely are performing different processing tasks. And of course, it depends on what is the goal of their cortical targets. So what the pre-processing here will affect their targets, and all of these layers have different targets. And so it's not surprising that there's some variety in processing, at least along the spectral-temporal modulation aspect that distinguishes these regions. So in other words, while best frequency, sort of simple parameter, may be columnarily organized, there's a specific variation in the modulation preferences across layers. Now back to a sort of bigger view, what happens to the spectral tempo modulation across the whole auditory hierarchy? So it has been known for a long time that let's focus on the tempo modulation range here for the moment, that the tempo modulation changes dramatically between elements in the eighth nerve, auditory nerve, and cortical neurons. So in the nerve, we can see that the modulation range that these elements, the axons or cells in the cochlear nucleus, can follow in time-locked fashion is much higher than in cortex, and it's a gradual decrease in the range that it's being represented by some kind of phase-locked activity. That is also true for within cortex itself. If we just look at primary fields here, here's an example from cat auditory cortex. We look at the tempo modulation frequencies in the core areas, A1 and AF. They are clearly higher than the modulation that is reflected in belt or parabelt areas. So this reduction in represented modulation frequencies is declining even in cortex itself in some kind of hierarchical fashion. Down here is an example from human parabelt areas, so recordings from the superior temporal gyrus using electrocorticography. And there we can see even lower modulation frequencies that are being kept and showing some kind of synchrony and phase locking. In this case, it's below 3 Hertz. So altogether, there's clearly a hierarchical reduction of temporal modulation that is phase locked in the response. The responses are phase locked to these modulations. Although, on the other hand, the spectral modulation range stays fairly constant. I'm not showing these data here, but if you look at just the parabelt in humans, the range of modulation, spectral modulation frequency here, still fairly broad and not much smaller than what you see in subcortical stations. So the question is, what leads to this reduction in temporal processing or spectral temporal processing across a hierarchy? So for that, we have to move to a different level of analysis at scale, namely to the synaptic level. And we have to apply some wholesale recordings to determine what are the inputs and outputs of a cortical neuron when it comes to modulation processing. So using voltage climb methodologies, we can identify excitatory synaptic currents to, again, we use our dynamic moving ripple stimulus as the input, and we can derive just for the excitatory synaptic currents, what is SDF and what is a modulation transfer function. We can do the same thing for the inhibitory synaptic currents, derive those modulation properties. With the current clamp approach, we can also look at the memory potential and the postsynaptic potentials that are driven by the synaptic inputs. And of course, we can look at the spiking moments or the temporal spectral modulation properties when a spike occurs. So by comparing these 4 stages of processing within a neuron, we see a couple interesting aspects. So first one, if we look at the best temporal modulation frequencies for these four signals. So for the excitatory synaptic currents, we see the highest average best temporal modulation frequency. This inhibitory currents have a slightly lower best temporal modulation frequency. But then when we go to the postsynaptic potential, it's clearly reduced. It's about half of that of the excitatory inputs. And spiking it reduces the best modulation frequency even more. So the clear decline from the input to the output due to the integration of spectral and excitatory and inhibitory inputs that create this transformation. We see something similar, or looks a bit the opposite for best spectral modulation frequencies. So the excitatory currents and the inhibitory currents have actually the lowest spectral modulation frequencies, and we know those correspond to the broadest tuning curves. But once we get to the PSPs, so the postsynaptic potentials and the spiking activity, the spectral modulation frequency increases a bit due to the integration of excitatory and inhibitory inputs that sharpen basically the tuning curve. So the principle here is that synaptic integration of excitation and inhibition result and a spike initiation process reduce the temporal modulation preference from input to output of a neuron, but increases spectral modulation preference from input to output of the neuron. So there's a change in the modulation preference that is induced at the neural level. So let's move on. So now we make a big jump in our discussion of modulation, namely, can cortex change its representation of modulation, spectral and temporal modulation through plasticity? So the cortex is a premier station that allows or permits a large degree of functional plasticity, certainly much more so than in subcortical stations. And cortical plasticity has been studied in many ways. And I just want to show you a couple examples that the plasticity, of course, applies to these temporal and spectral modulation properties as well. So here's an example where we trained cats to do a simple task and a more difficult task that relates to the spectral envelope processing. So the first task here in A is just a simple lateralization task. Cats can do it very quickly and very accurately, of course. So the stimulus is a broadband stimulus with a spectral modulation envelope. In this case was 2 ripples per octave for the fairly narrowly spaced peaks. The other experiment is more difficult for the animal. The signal comes from the front, and it's again a spectrally modulated signal. In this case, with 1 ripple per octave. But the 2 signals that the animal has to discriminate have different ripple phases. In this case, in the illustration, they're 180 degrees out of phase. So it takes them a while to learn to make that distinction, but they learn it eventually very well. So now what happens to the cortical modulation preferences for spectral modulation? In naive animal here, that's the blue curve. This whole thing is a ripple transfer function with the firing rate versus the density or the spacing of the peaks in the stimulus. In naive animals, the highest firing rate is around 1/3 octave to 1/2 octave spacing of the modulations. But when we look at these 2 trained animal groups, we notice that for the 2 ripple condition, there's a big increase at that ripple frequency in firing rate. And for the other case, there's a big increase in the 1 ripple per octave ripple density plot. So clearly, the cortex has picked up the important stimuli and improved their response, as we would expect in a highly plastic system. The function behind it, how that works, is likely related to sharpness of tuning curves. And here's just, it shows that the sharpness of tuning measured as Q40 is lower in the naive animals, and tuning gets sharper in these two trained groups. So the principle here is, of course, that spectral modulation preference is enhanced for rewarded spectral modulation stimuli. Here's an example from a temporal modulation plasticity. So in this case, animals were trained, these are all monkeys, in a temporal discrimination task. So the animal listened to two sequential stimuli. The first one was a standard, always a tone or a signal modulated at 4 Hertz. And then the target was at modulated higher temporal modulation frequencies. The animal had to say when the higher modulation occurred. We also did it with the 10 Hertz as target, but the result was really the same. So what happened in the cortical neurons once the animals have been trained? Up here in this plot, we can see that these are the modulation frequencies in naive animals, or the distribution or the modulation transfer functions for tones for temporal modulation. You can see that there's the strongest response, its strongest phase locking is around 8 to 10 Hertz, and then it drops off, and higher modulation frequencies don't produce a strong phase locked response. However, if we look at our trained animals, we see two changes. First, this big, strong response to these low modulation frequencies is suppressed. This is around that standard stimulus, which is used a lot, and the response is being reduced. But on the other hand, the higher modulation frequencies are greatly enhanced in their ability to phase lock. This is here the population plot, where we can see the naive animal have this low pass characteristic for which modulation frequencies are preferred, whereas the trained animal clearly shows a big emphasis on these high modulation frequencies in their ability to phase lock. The second experiment shows really the same result. So temporal modulation processing is enhanced for rewarded stimuli, these high modulations in this case, and is reduced for not rewarded stimuli, the standard. Just one quick aside. So why then do cortical neurons prefer these low modulation frequencies? Because it's not a limitation of cortical neurons can do. This clearly demonstrates cortical neuron can phase lock much higher than what is normally in the naive animal being represented. So that's an interesting question, because it may have a key to what cortex is actually trying to accomplish. And these low modulation regions that are often seen being responded to seem to contribute an important facet to whatever problem the animals want to solve. Okay, now we move to an even more complex theme, and that is we want to look at how we can improve the characterization of cortical neurons over the standard approach that we have used so far, that is widely used, namely that of spike-triggered averaging. So spike-triggered averaging is a linear approach and has really provided great insights in what cortex and subcortical neurons are doing. But there are a number of studies that indicate that that approach provides an incomplete characterization of neural function. So what to do? One of several approaches that utilizes nonlinear methods to reconstruct receptive fields is called for. So we use a method that is sort of quickly illustrated up here, that it's not an averaging of a signal preceding a spike, but it fits a filter or two filters, actually, so that the mutual information between stimulus and the spike train is maximized, and that is achieved by pixel adjustment of these two filters until some kind of maximum is reached in the mutual information. And that gives us then two filters that help describe what the cortical neuron actually responds to. So these are called most informative dimensions, number 1 and number 2. So what are some of their properties? So here, compared to the linear approach, this is the spike-triggered average that we usually use. The The first MID1, the first filter of this other approach, is actually very similar in shape and in characterization. So we capture much of what the SDA is doing, but actually we capture it in a better way because the mutual information captured by the MID1 is significantly higher than what is captured by the spectral average. But sort of the more interesting aspect is really in the fact that we have a second filter that we can utilize that has actually a different shape, almost by definition, it cannot be the same. And you can see extra sidebands and longer stretches of activity that seem to be associated with the occurrence of a spike. So the information that is captured by MID2 is much smaller than MID1. But these two filters act in concert. That is, jointly, they actually capture more information than if they are tested separately and independently. So the sum of the independent filters in information is less than what the two filters combined will provide, which is expressed as synergy between the filters. There's some kind of interaction that boosts the total coded information when they both are involved. And the synergy is particularly expressed in cortical stations. Here, this is the amount of synergy, 100% is no gain, where here we can see that A1 and AF have a significant gain, whereas the subcortical stations, synergy doesn't really do much. And we already mentioned that the MID2 in those subcortical stations is usually quite small. So now we can identify, we gain a maybe better description, a more complex description of what cortical neurons might be doing. How does it relate to our quest to understand the spectral and temporal modulations? So here's an example of a neuron in rat A1, where we determined the two filters, MID1 and MID2, also shown here. And these are the modulation transfer functions of these two filters. And you can clearly see that they're not identical. So the transfer function for the MID1 has a higher temporal modulation frequency and a higher spectral modulation frequency than what we see in the MID2. So in other words, and that is true for the population as well. So the MID2 modulation response occupies a different range than for the MID1. So in other words, these two filters look at the same time, in a way, at different modulation properties within the stimulus. And if they are just right, they somehow support each other and create a more synergistic response to this combined spectral temporal modulation response. What is sort of intriguing a bit, it's a speculation, but nonetheless, let's speculate a little bit. If you remember, we looked at the parabelt regions in humans for their spectral and temporal modulation preferences. So here it is shown again, and we notice that most of the modulations are below 3 Hertz. So it's fairly low temporal modulation over a fairly wide range of spectral modulation. Now, if you look at this little red box around it and superimpose it here on these modulation spectra of the MID1 and MID2, clearly there's a mismatch here for MID1. It's a much lower region, but here for the MID2, actually, the match is not bad, indicating maybe, as a speculation, that what determines the MID2 is really related to some kind of feedback from higher cortical stations. That is particularly interesting because the latency of these MID2 filters is significantly longer than that of the MID1. I mean, there is MID1 response, 10 to 30 milliseconds after the stimulus, whereas MID2s have latencies of 50 to 70 milliseconds. So there's some kind of time delay when the MID2s are being expressed at that neuron. And so there's certainly the time there that could provide this feedback or contextual information that may help to modulate what MID1 is actually processing. So our principles here are that MID2 modulation preferences are lower than for MID1, and potentially at least MID2 may enable a sort of a context-dependent or feedback-dependent processing for the first filter. So moving on to our last topic, and that is, so in the what we talked about so far, we really look mostly at what a single neuron is doing. Now, when we record now, we are able to record from many neurons simultaneously. And what we find is, not surprisingly, that many of the activities that we record at the same time show some type of correlation with each other. So there are two types of correlation patterns that can be observed between two neurons recorded at the same time. And we are still talking about recordings within the column of across the depth of auditory cortex. So here are a couple examples of two STRFs within the same penetration. And here we look at two correlations. One is between the shapes of the STRFs. So we just correlate this shape with that shape, and that gives us this outline here, this sort of sinusoidal looking thing. So that tells us how much overlap is there between the two receptive fields. But the spiky inlays, those are the correlations of the spike trains of the two neurons. And in this case, those two correlations actually look quite similar, and which would indicate that the spike train correlation is really a consequence of the similarity of the two receptive fields that respond to stimuli, to the same stimuli, but they do it in independent fashion. They're not connected or anything. They're just responding to the same stimulus with their own spike train. And that's what reflected in this fairly broad correlation between two neurons. On the other hand, we see often, excuse me, neurons that have a very sharp correlation function. So here's an example of another pair that compared to this receptive field correlation, which again is this sinusoidally looking line, have a very sharp correlation or show some kind of synchrony in their spike occurrences. And that could be either that they receive inputs at the same time, or actually that there is a connection between these two neurons. So now these, it has been proposed that these more sharply correlated neurons forming what's called the cell assembly, or we call it coordinated neural ensemble, meaning that these neurons have a tendency to fire together, and that might improve the information that is being conveyed by this group of neuron to the next station. And so what are those properties with regard to modulation and so on of these synchronized events in auditory cortex? Actually, there has been a suggestion. I mean, these ensembles have been observed in other sensory cortices in the hippocampus. And actually there's sort of a movement going on that wants to label that these ensembles are really the basic functional organization of cortical processing and not necessarily the single neuron, which is sort of evolved, so to speak, from the single neuron doctrine as the main carrier of information. So these group of neurons somehow seem to be relevant because a couple short points about this. The first thing is the activity that these ensembles respond to has a higher precision. It carries more information than what individual neurons are doing. So here in the lower left is an illustration of that. So we look at a single neuron here in this upper plot that responds to a segment of our ripple stimulus. And you can see, you can sort of guess that there are some regions, some times in this response where the repeated stimuli tend to respond more often than in between. Here's the same thing now for this group of neurons so the event of the ensemble. And here you can see a much cleaner phase locking, so to speak, to that stimulus segment, which indicate that the mutual information between spike transient stimulus is lower here for the individual neuron compared to the ensemble event. And that it can be shown that the mutual formation ensemble events is clearly significantly above the individual neurons contribution. So ensemble events present sort of a more cleaner version of what the individual neurons can provide. It sort of strips out some noise and unwanted or unrelated activity. The other aspect of these ensembles is if you the old adage of you fire together, you wire together. So it is potentially possible that these neurons that fire together target the same neuron or the same locations in the next station that is being projected to, and will improve the transmission of information from one station to the other. Because they work together, and synchronized activity has a higher probability of eliciting a spike in the next station. So those are a couple of reasons why these ensembles are of interest. Quick question regarding our modulation properties. So here in the upper right, we see an ensemble the way it has been analyzed. So this analysis is based off, or what determines what belongs to an ensemble is based on a principal component analysis followed by independent component analysis. And here we see the independent component weights for 14 neurons in penetration. And these three have been statistically determined to be members of the CNE. Now, if you look at the modulation properties of these events, they look pretty much like those that we are used to from individual cortical cells, but perhaps they are cleaner. They are more restricted in the preference in this spectral and temporal modulation range. So one sort of intriguing observation that we made recently is that occasionally we see these double CNEs, which are demonstrated here. So first, here are all these individual weights of these neurons with regard to the joint synchrony. And we see actually two groups of neurons that qualify to form an ensemble. Here, the red ones, and on the other end, the blue ones. Now, they have different sign of the weights. That means that they're really different. They're not belonging, they are not firing together. But on the other hand, they're somehow temporarily linked. And it turns out that these two ensembles that we can identify don't respond at the same time, but they really sort of respond mutually exclusively. That is either one or the other, indicating that there might be some kind of link between these two, but they only enable one of them to respond at any given time. This could be achieved by some kind of cross-inhibitory mechanism. And indeed, if you correlate activity of one neuron in this group with another group neuron in this other group, you do see sort of a negative correlation function here. This is what is shown, correlation coefficient between the blue and the red, and this population here. And we can see there's a large negative correlation when one of these fires and the other one will be silent. So there's some kind of linking between two different ensembles. And we don't really know what this cross inhibition actually will be doing. So we have to still study that. But this ensemble approach just offers another window on another scale of cortical processing that may influence what we learn about auditory cortical processing, whether related to modulation or to other, its utility to other aspects of creating perception. So just the principle, and quickly here, the coordinated neural ensembles activity carries more information than its member neurons. The ensemble activity improves the modulation and coding, quality, and transmission. And potentially, at least, interacting ensembles cover mutually exclusive modulation ranges and may reflect some cross-inhibitory mechanism. Now, what else are these ensembles doing? We will come to that later, but we are actively working in collaboration with Andrea Hasenstaub to elucidate some of those aspects. So let's summarize now what we have learned or what you have heard. So the goal was to look at different functional structural levels. What could each of those different levels contribute to our understanding of modulation encoding, spectral and temporal modulation encoding? From at the synapses, we learned that excitatory and inhibitory synaptic currents have, on average, higher temporal and lower spectral modulations than the postsynaptic potentials and the spiking that arises from those inputs. From at the cellular level, we learned several things like cortical neurons prefer lower modulation frequencies than subcortical cells. We also learned there's sort of a general trade-off between the temporal and spectral modulation frequencies, meaning neurons that have high temporal modulation usually have low spectral modulation preferences, and vice versa. We learned from our two-filter approach, or more complex SDF approach, that they're cooperating filters, but that they really focus on different modulation properties. Exactly what that means for the processing of sounds isn't clear yet, but it may reflect some kind of influence of context or feedback on the processing of that modulation information. The circuit and ensemble level, we learned that there's more information carried about modulation by these ensembles than their individual member neurons. And again, speculating here, interacting ensembles may have an interesting relationship between their modulation properties that still needs to be clarified. From looking at layers, we noticed that the modulation preferences across layers, even within the same column, is not uniform. So each layer is sort of doing its own thing. And that's not surprising because each of them has their own target and their functional purpose, which is related to what their output goal and the output target might be. At the even larger level, we looked at modules and found that spectral integration modules in A1 are not only functionally defined, but also anatomically defined and create some parallel streams of different modulation properties. And then at the larger field level, we learned that the core receptive fields prefer higher tempo modulation than belt and parabelt fields. Although on the other hand, spectral modulation preferences are much more uniform across different stations, maybe with a little reduction in the parabelt. So it's all different aspects that we glean from looking at these different structural scales of information processing. Quick word about plasticity. So clearly both spectral and temporal modulation properties can be altered by experience through training, which is not surprising, given that auditory cortex is such a hub of flexibility and cortical mechanism that can change what is being encoded. What was sort of interesting, at least to me, was the experience-induced plasticity of higher tempo modulation frequencies in cortex. So the boosting of the higher modulation frequencies after training is sort of in contradiction to what normal auditory cortex is representing. They usually focus on modulation frequencies below, let's say, 20 hertz or so, whereas the training can enhance these higher modulation frequencies. So, but that indicates that this focus on tempo modulation frequencies below 20 is not due to some kind of structural limitations like timing jitter or because it's a tense synapse from the cochlea or something, but it's really seems to be a choice of the cortex because that modulation range helps it to better process specific functional tasks that we actually don't know what they might be. But if we think about modulation processing as related to speech, it makes great sense to look at these low modulation frequencies because that's where much information in these complex sounds is. So the overall verdict then is more complete understanding of cortical function really requires knowledge at each of these structural scales from very small to very large to really figure out what is going on. So just a quick look at future topics we are interested in. So how do our cortical neurons work together in these ensembles, and how do they encode sensory information, and how does it influence behavior? So in other words, what do these CNEs actually good for? So we have some guesses. It has something to do likely with the robustness of sensory noise, so that there's some kind of creation of invariances, whether it's for signal noise or just for signal without noise. That there's improvement of robustness compared to neural noises that might appear through many of the inputs. There's also maybe a selective improvement of information transfer from one station to the other, like from MGB to the cortex, or from A1 to belt and parabelt regions that are being enhanced by the utilization of these ensemble activities. Also, how ensemble activity relates to behavioral demands in a task, and how they vary and adapt to that those are all open questions. We also look at cortical encoding properties in hearing loss in large animals, actually, especially in symmetric hearing loss, to see an animal with similar hearing properties in humans. What can we learn about the impact of moderate to mild hearing loss on cortical organization? But there are other broader questions about behavioral molecular changes in hearing loss, which can be studied in rats and mice. There are also opportunities from extending our technical repertoire that will enable us to have a much better idea of how different cell types contribute to cortical processing. That is just in its infancy to learn more besides the fact that they're excitatory and inhibitory neurons. We can also study more about the cell ensembles, and through cell manipulations learn more how local circuits perform their duties. So we are recruiting for open positions. Everybody interested, please contact me or Andrea. So final words here. So I want to thank really all my collaborators, and here is most of them. I've been really lucky to have found so many gifted, productive, and congenial collaborators. As I said before, this is a team effort, and all of these people are deserving to partake in this recognition. So therefore, I have a special thanks for all of them for helping to push and pull the wagon through the mud of scientific uncertainty. So thank you, everybody, and thanks for recognizing our work. Okay.
Christoph, what a wonderful talk. Thank you so much. It just shows how much of a team effort science is, and yeah, you were lucky to have great collaborators, but I think you also did tremendous work in bringing people together. You really meet the criteria of the ARO Award of Merit for sustained scientific achievements and contributions to our field. So congratulations. And I do look forward to having dinner with you at the next in-person ARO and other council members and whatnot, because you deserve it. And I'd like to talk with you more about this. Okay, just to kind of close things out, I just want to remind everybody the nominations are still open for the ARO 2023 Award of Merit. We're not going to select the next winner until 2022, or sorry, this summer, I guess. And so the nominations are open until May 31st. Nominations may be submitted by any ARO member. We strongly encourage diversity among both the nominators and the nominees. So please think about people that you might want to nominate and just go ahead and do it. And I think it's time to close out this session. Take a 15-minute break.
Can we ask a question about that?
Oh, yes.
Why?
Why what?
Why is people that nominated people obeyed one deadline, and now you're changing the deadline, I don't understand. Did you not get good enough people awarded? I mean, nominated?
Oh, no, we
Or-
had great nominees. We are looking for a lot more diversity in the nominees.
So is there some rule about diversity if we need to, the council has decided? I always thought this was a kind of lifetime achievement award.
It is. I might refer you offline to speak with Ruth Latofsky, and who kind of led some of this effort. I'm a little bit out of the loop on it, but it's probably, we don't need to get into it right now. Ed,
Thank you.
thank you. Okay, well, thank you very much. Let's take a 15-minute break and move back to the meeting at 11.
Thanks, everybody, and congratulations again to everyone.